In this presentation, we're going to look at another one of our biomacromolecules, and this time we're going to look at nucleic acids, whereas obviously so far we've been looking at proteins. And similar to proteins, what we're going to look at is the structure of different nucleic acids, have a little bit of a look at the bonding types of reactions that take place, and then eventually what we're going to do, not in this slide, but in our next sort of topic, is really nail that connection between nucleic acids and proteins. As I said, nucleic acid is a biomacromolecule, which means it's a large molecule composed of smaller molecules. So we'd say it's a polymer, and the monomers of a nucleic acid are called nucleotides. So the monomers of a protein were called amino acids. The monomers of a nucleic acid are called nucleotides. So with nucleic acids, that's a blanket term. So nucleic acids, there's two types. We've got our deoxyribonucleic acid, DNA, or our ribonucleic acid, which is RNA. Both of those nucleic acids, though, they're both polymers, and they're both composed of the monomers called nucleotides. When we're looking at DNA and RNA, their roles are similar in the sense that they contain instructions, genetic material, that plays a role in the synthesis of proteins. Here's a slide with a few questions for you to think about. I won't go through the answers here, but you can do this with your teacher, which is me, or Miss Hunter or Mr. Van, back in the classroom. So where is DNA located? What does DNA do? On question one there, where is DNA located? Most of you are going to say the nucleus. There's a couple of other areas too where you could find DNA that you should have a little bit of a think about. Where does an individual's DNA come from? What does a DNA molecule look like? What are the nucleotides of DNA? How are the complementary strands of DNA held together? And these are all things we're going to answer as we go on. Let's look at the structure of DNA. What we've got here is a diagram of one strand of a DNA molecule. So up the top, I'm going to draw a simple diagram. I'll try and draw two simple diagrams. So you might think of DNA as that double helical molecule that it is, that sort of looks like that. If I draw another simple diagram of that, so I'm drawing the exact same thing, but let's just say I'm representing it with two chains. So what DNA is it has 1, 2 nucleic acid chains. Those chains are bonded together and then they coil up to form this structure over here, which is the double helix. So what I'm drawing, what we've got down in this diagram, is just a snapshot of one of those chains. So the chains are identical in the way they're made. So it's fine for us just to look at one of them. So let's have a look. Each chain in its own right is a polymer because each chain is composed of lots of monomers joined together. And you can see on this diagram, you can see we've got the monomers, which are called nucleotides, and this little box here has outlined one nucleotide. So one nucleotide has a sugar, and this is DNA, so it's a deoxyribose sugar. It has a phosphate, and it has a nitrogenous base. And in this case, because it's got the letter A, I know that that base is adenine. So a nucleotide has a sugar, a phosphate, and a nitrogenous base. And essentially, to make a nucleic acid, we just take lots of individual nucleotides and we join them together. So in this diagram, we've got 1, 2, 3, 4 nucleotides joined together. You can see where they do join. So the phosphate here of that nucleotide is joined to the sugar of the nucleotide before it. And then as we go down, we always join sugar to phosphate, back to sugar, back to phosphate, and what we call this line down here, going all the way down, is a sugar phosphate backbone. So it's like the backbone of the molecule made of sugar phosphate bonds. And each of those bonds between the sugar, well, between two nucleotides, so a bond between two nucleotides, is called a phosphodiester linkage. If I was in the classroom with you right now, I would say, can you remember what the bond is called between two amino acids? And you would say a peptide bond. Excellent. This is just the equivalent, I suppose, but this is what it is when it's nucleic acid. So the bond between two nucleotides is a phosphodiester linkage. Like forming a peptide bond, though, it is formed through a condensation reaction. So we join one nucleotide to another through a condensation reaction that forms a phosphodiester linkage. So I could ask you the question as well, looking at our 1, 2, 3, 4 nucleotides joined together here, how many molecules of water would be released in forming these 4 nucleotides joined together? And you might say 1, 2, 3, because we've got 3 phosphodiester linkages. This slide here just goes over some of the stuff we talked about on the previous slide. So it's that same diagram. It's showing you in red the phosphodiester linkages, which we just learnt were formed through condensation reactions. And the important thing to remember is that that phosphodiester linkage is a type of covalent bond, just like the peptide bond in proteins is a covalent bond as well. A covalent bond is a really strong bond, so each nucleotide in the chain is joined to the nucleotide above and below it quite strongly through that covalent bond. Over here we've got our bases. So as we said before, we've got 4 different colours here representing the different bases of DNA that we have. So we could have adenine, which is our A, thymine, which is our T, guanine, which is our G, cytosine, which is our C. So we have 4 different bases. Essentially, if you were looking at a DNA sequence, it'd just be a big long line of the letters A, T, G, and C all being repeated in different sort of ways. We need to be able to read that sequence, and in your body you have some enzymes that need to be able to read that sequence to make some messenger RNA to then go on and make a protein, which we'll talk about later. It's a complex process in its own right. One thing to remember for now is that if you were looking at, let's say, 100 A, T, G, and Cs all repeated in a DNA sequence, there is some order to the way we read that, and they're grouped into what we call triplets. So we always read a DNA sequence in groups of 3, sort of starting from left to right or top to bottom in this example. And what you'll learn down the track is this group of 3 nucleotides will relate to a group of 3 nucleotides in an RNA called a codon, and those 3 nucleotides will relate to a group to a specific amino acid, but more on that later. Just like with proteins and amino acids, we had a closer look at what an amino acid looks like. This slide's giving us a closer look at what a nucleotide looks like. And that's definitely something you would need to be able to draw and label for yourselves. Definitely a diagram you would need to be able to recognize as being a nucleotide. For example, if I gave you a diagram of a nucleotide and one of an amino acid and didn't tell you what they were, I would expect that by looking at those diagrams, you can say which one's an amino acid and which one's a nucleotide. So as I said before, the nucleotide's got the 3 main regions. It's got the sugar, it's got the phosphate up the top, and it's got the base. And you need to know which parts of the sugar that base is being attached to and which parts of that sugar that phosphate is coming off. For every single DNA nucleotide, they all have deoxyribose sugar, they all have a phosphate, and they'll have one of 4 bases: adenine, guanine, cytosine, or thymine. If it's an RNA nucleotide, it's a little bit different. The sugar would be ribonucleic acid, the bases would be adenine, guanine, cytosine, but they would have uracil instead of thymine. With our bases, our 4 bases, we can further categorize them as purines or pyrimidines, which refers to how many rings they sort of have in their structure. So there's slightly different structures, which we'll look at in a minute as well. But you want to find a way to remember that adenine and guanine are purines, and cytosine and thymine are pyrimidines. What this slide's showing you here is that, as I said before, we've got our bases: adenine, guanine, thymine, cytosine, and you can see uracil down here. That one's only in RNA. We can classify our different bases into one of 2 groups. So we've got 2 groups here. One's called the purines, and the other one's called the pyrimidines. And the difference is in the ring structure. So if you look at the purines first, you can see that adenine is a purine and guanine is a purine, and they both have 1, 2 rings fused together. 1, 2. If you look at the pyrimidines, thymine, cytosine, uracil, they only have a single ring, and that's the difference. Don't see too many questions on this really in a VCAR exam or in your SACs, but it is an important thing to try and remember. I always try and think of crazy little ways to remember all sorts of things. This is one of my weirdest, but I'll tell you anyway. It might work for you, or you might just think that's ridiculous and come up with your own way. But as you guys would know, we have the base pairing rule. So I think you would remember from year 11 probably that with bases in DNA, A always pairs with T and G always pairs with C. The way I remember that, I think back to when I was at uni and I did my uni on the Gold Coast, and I always, just for some weird reason, I think at Gold Coast. So at A to T, Gold Coast, G to C, at Gold Coast. The other thing I think of is if you say purines and pyrimidines, purines, pyrimidines, purines, pyrimidines, like I feel like that flows off your tongue. But if you say pyrimidines, purines, pyrimidines, purines, pyrimidines, purines, it sort of doesn't flow. It's more natural to say purines, pyrimidines. So I also think at Gold Coast, A is the first letter and then G is the first letter. Purines, pyrimidines. Purines are like the first thing in what you say. So I think the A and the G, the first letters, match with purines, which is the word you always say first. That's how I remember it. Another way you could remember it if you wanted to is you could think purines is the smallest word, yet it's for the larger molecules. Pyrimidines is the longest word, but it's for the smaller molecules. I might leave that one with you. This slide here is showing you 2 scientists down the bottom here, James Watson and Francis Crick. So sometimes you hear the Watson-Crick model, or Watson and Crick. And another thing you guys will have heard before is that DNA is referred to as a double helix. So double meaning 2 strands, helix meaning it winds up into this helical sort of shape. So these guys were the scientists that made this conclusion that DNA was a molecule that had this particular shape. And the reason DNA has this shape is from the way the 2 strands bond and interact with each other. So what we know by the double helix is that DNA is double-stranded. We know that each strand is alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, which we call our sugar phosphate backbone. We know that the two strands are held together through hydrogen bonds, which are weak bonds between adenine and thymine, A to T, at, and guanine and cytosine, Gold Coast, G to C. And the fact that they're hydrogen bonds obviously tells you guys that they're weak bonds. So DNA can come apart and it can go back together quite easily too, which is a really useful thing when we need to replicate it. These next two slides are pretty simple to follow. Well, simple to follow, but could be confusing in video form. Probably easy if I show you it on the board when we're in class. But it's just a simplified diagram of the double helix. So down the bottom on the right, we've got a double helix all twisted up. And you can see in the middle what we're showing there is the T and the A pairing, the G and the C pairing. So that's our hydrogen bonding between the complementary bases. This is just showing that exact same double helix molecule, but not in its helix form. So you've got your single strand here, your other single strand here, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate backbones, bases facing the middle, and showing that their hydrogen bonds occurring between the bases. This slide's just a nice little follow-on from the one we were just looking at. It has a little bit more detail in it. What this one tells you is that the way the strands are aligned, we say it's called anti-parallel. So obviously parallel is when two things are running in the same direction side by side. Anti-parallel means they're running in opposite directions, but they're side by side. So you'll notice on this picture, the strand on the left, we've got a phosphate being the outermost part, whereas the strand on the right, it's the sugar. That's the outermost part, and then it's the opposite at that end down there. So we say DNA is anti-parallel, and we name the ends. So the end where the phosphate is facing out here, we call it the 5' end. The end where the sugar is facing out, we call it the 3' end. And because they're always opposite to one another, if the 5' end is on one strand, then directly opposite that is the 3. At the other end of the 3 is the 5, opposite that would be the 3. The other thing this diagram is showing you is just emphasizing that those bonds between the sugars and the phosphates are covalent bonds. So really strong bonds between the individual nucleotides in each strand, but weak hydrogen bonds between the bases holding each strand together. Just a reminder down the bottom, we've always got what we call our base pairing rule, complementary base pairing rule. Adenine always joins with thymine, guanine always joins with cytosine. Here's a little question for you, one that you might sometimes see in a SAC or an exam. So the best thing for you to do is pause me and then have a go at it, and then unpause and I'll keep explaining. So pause now so you can have a go. Let's imagine you've come back. So we've got a strand of DNA was analysed. It was found that it comprised 35% of cytosine bases. The first thing you're going to do with this question is think about what concept is it trying to get me to demonstrate that I understand. And what it wants to see is if you understand the base pairing rule. So we know that A always joins to T. Really need to get a stylus, don't I? G always joins with C. So adenine always joins with thymine. Guanine always joins with cytosine. So if that molecule's got 35% cytosine, well, those cytosines must be paired with the same amount of guanines. So therefore it must also have 35% guanine. 35% and 35% makes up 70%. So of the 30% of bases that's left, half must be A's and half must be T's. So 15% adenine, 15% thymine. So our topic is nucleic acids. And as I said in the very first slide, nucleic acids is a blanket term. So nucleic acids are a type of biomacromolecule, just like proteins are a type of biomacromolecule. Nucleic acids incorporates two different molecules. So it incorporates DNA, deoxyribonucleic acid, which we've been talking about up until this point. That's our double-stranded, double helical molecule. But the other type of nucleic acid is ribonucleic acid, which is RNA. And we haven't talked about that one too much yet. So it's important that we take a bit of time now to distinguish it from DNA, and then have a little bit of a chat about what its roles are in a cell. Also, a relatively common type of question for you to get would be to compare some structural differences, or even functional differences, I suppose, but definitely structural, between RNA and DNA. And if we just look at these two pictures here, so the DNA one on the left, the RNA one on the right, first thing you can notice is DNA is two strands, RNA is a single strand. So that's your first structural difference. RNA is single-stranded, whereas DNA is not. Nice reminder for you too, if you're ever answering a question like this, a compare question, make sure you talk about each thing. So give each thing its own sentence. So for example, if the question said, identify one structural difference between DNA and RNA, and let's say you wrote RNA is single-stranded, full stop, you wouldn't get your marks. What you would need to do is say RNA is single-stranded, whereas DNA is double-stranded. Super important. There's three main structural differences, so that's the first one. The other structural difference is in the bases, the nitrogenous bases. So in DNA, we have adenine, thymine, guanine, cytosine. In RNA, we have adenine, but there's no thymine. No thymine in RNA. In RNA, the thymine is replaced with a different base called uracil. So in RNA, A joins with U instead of joining with T. So instead of saying at Gold Coast for RNA, I just say like AU, like AU, what's going on? So A to U, and you can still keep Gold Coast, that's fine. So there's our second difference. If we were putting that one into question form, comparing, question was name a second difference, you'd say RNA contains the nucleotide uracil, whereas DNA contains the nucleotide thymine. Our third difference, you can pick it in the name. So we've got deoxyribonucleic acid or ribonucleic acid. So I didn't mean to do that. Ribo, deoxyribo. And what that's referring to is the type of sugar that it has. So if you think of your structure of a nucleotide, you have your sugar, your phosphate, and your nitrogenous base. In DNA, the type of sugar is a sugar called deoxyribose sugar. In RNA, the sugar is one that's called ribose sugar. So those are our three differences. RNA is single-stranded, RNA has uracil instead of thymine, RNA contains a ribose sugar instead of the deoxyribose sugar. With RNA, it's not enough for us just to say we have DNA and we have RNA. We actually have three specific types of RNA, and it's super important that we understand. We can name each type, we can identify what each type looks like, and that we can briefly explain the role of each type. And because they all work together to help us make a protein, we'll define them here, but when we get into doing transcription and translation, we'll look at their role in a lot more detail there. So three types of RNA. we have a type called messenger RNA, which you often see abbreviated as mRNA, so little m RNA. Our second type is ribosomal RNA, which we call rRNA. And our third type is transfer RNA, which we call tRNA. Just remember, it's transfer, not transport. Often people do make that mistake. So 3 types of RNA: messenger RNA, ribosomal RNA, transfer RNA. Really basically what their function is. So we'll talk about mRNA first, which is a diagram up here. So mRNA is transcribed from DNA. So let's say it's made from DNA. And it carries a genetic message to a ribosome. So DNA has the instructions to make a protein, but we need to take those instructions, transcribe them onto a length of RNA, mRNA, and then that length of mRNA can exit the nucleus and travel to a ribosome, because of course the DNA never leaves the nucleus. rRNA, its role is to actually form the actual ribosome. So rRNA combines with some proteins. So if you guys are thinking back to what you know about proteins, you could probably say the proteins in a ribosome are conjugated proteins because they're associated with a different type of molecule, being RNA. So rRNA literally forms a ribosome by bonding with some proteins. Transfer RNA. So you guys would know that at a ribosome, we make proteins. You'll also know that a protein is essentially lots of amino acids joined together. So we need to get the amino acids from the cytosol of the cell to the ribosome so that at the ribosome they can get joined to each other. That's the role of tRNA. It attaches amino acids to itself and then it transfers them down to a ribosome, drops them off at the ribosome so that they can get formed into a protein. So all those slides up until now gives us a pretty good outline of sort of structure and function of DNA as a molecule and then RNA as a molecule. And previous to that, we also looked at proteins as a molecule and sort of what their function is. These next few slides, what I want to try and do is explain how they link together quite basically, knowing that it's a topic in itself where we're going to break down this process and look at it in more detail. But what I want to drum into your heads for sure is that there's a link between DNA and proteins. DNA has the instructions to make proteins. Proteins carry out all of those different functions in your body, make up the structure of your body, etc., etc. So what we refer to as the central dogma of genetics, so this is like the main thing about genetics, is that your DNA has sequences of nucleotides along it that are organized into things called genes. Each gene has the instructions to make a specific type of protein. The thing is, DNA cannot exit the nucleus, yet proteins are made at the ribosome. So DNA can't pass a message directly to the ribosome to make the protein. It needs to pass it to something that can exit the nucleus. So we start with the instructions on our DNA. A process called transcription takes place, which forms a length of messenger RNA that contains a copy of those instructions. The messenger RNA leaves the nucleus and goes to a ribosome. At the ribosome, the message on the mRNA is translated into an amino acid form, which causes specific amino acids to attach in a specific order to one another at the ribosome, and that forms our polypeptide. And as you guys know, that would then go off and get folded up into a functioning protein. So take from that, a gene is literally just a sequence of nucleotides that have the instructions for a specific protein. And there's thousands and thousands, I think 20 to 25,000 different genes in the human body. And every single cell has the same copy of DNA. So you've got 20 to 25,000 different genes in every single cell in your body. They're not all turned on though, and we'll talk about that later. DNA is stored in the nucleus, yet a polypeptide is formed out in the cytoplasm at a ribosome. So messenger RNA is like our middle person, takes a copy of that message and delivers it to the ribosome. And then what we've finally got, and we'll talk about this in a couple of slides, is the genetic code, which is this very specific code, which we display in a table, that shows you a group of nucleotides and how different groups of nucleotides relate to a specific amino acid. Let's continue. Hopefully not too confused, but I'll be pretty surprised if you weren't decently confused at the moment. A hard topic to understand through a video recording, so I'll definitely go over it in class, really simple for you. So I finished by saying mRNA, the nucleotides are sort of organized into groups, and each group relates to a particular amino acid. This picture might help explain that a little bit more. So let's do it in order. So here we've got our DNA. And what we remember from the slide just before is that the DNA has a set of instructions which we transcribe into a length of mRNA. mRNA. So where this arrow is, that process, if it was included, is, I can't write that, it's going to take forever. I write trans, which doesn't help because it could be translation too, but it's not. It's transcription. Got that? Transcription. So through transcription, we've made this length of messenger RNA. This length of messenger RNA has a sequence of nucleotides. Now, if you were to look at it, you could easily think what a confusing random sequence of letters. But there's order. And the order is that each of the nucleotides, sort of read from left to right, are arranged into threes. So we always read them in a group of three. So we read ATG together as a three, CTA would be together as a three, GGC would be together as a three. And what I'll show you in a moment on the genetic code is that each three nucleotides on mRNA and a group of three is called a codon. So on this example, we've got three codons because a single codon is a specific group of three nucleotides. Each specific group of three, each codon, has the instructions for one particular amino acid. So our amino acids here, let's say call this one A, this one B, this one C. So when the ribosome's translating the codon ATG, tRNA is always going to bring the A amino acid down. When the ribosome's translating the codon CTA, it's always going to bring down the B amino acid. GGC codon, it'll always bring down the C. So by doing that, what we're doing is we're following the specific order on the mRNA, and that's dictating the order in which we align our amino acids. This process where we go from mRNA to amino acid, that's the one we call translation. I won't bother writing that, eh? Translation. This is what we call the genetic code. So any question you'll get that requires you to link an mRNA sequence, a codon, to a specific amino acid, it's always going to give you this genetic code. Now, there's a few different ways you can present the genetic code, but it all means the same thing. So you don't need to remember all of the different codons. You don't need to remember all of the different amino acids. You just need to know how to read this table. And this is how you do it. So on the left-hand column here, we've got the first letter of the codon. Across the top, the second letter of the codon. Down the right-hand column, third letter of the codon. So let's say, for example, the mRNA strand at the ribosome, it is reading the codon U, U, U. And the way we're working that out is U is the first letter, U, so U is the first, U is the second, U is the third. So if it's reading the codon U, 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 then we know it is going to attach this particular amino acid here, which is PHE, which is phenylalanine. If it was, well, let's do the examples over here. If it was reading actually, I'll give you another example before we do the questions. If it was reading the codon CCU, then it would attach the specific amino acid PRO, which is proline. Let's try our questions. Here'd be a good time for you to stop and pause and try them yourself, and then unpause me and I'll go through the answers. So pause, unpause. Identify the codons of the following amino acids. So the amino acid CYS, which I know is cysteine. All we need to do is look for CYS on the genetic code. And I can see CYS is up here. So identify the codons that relate to that specific amino acid. You can see that there's actually two. UGU is a codon that would result in cysteine, and UGC is a codon that would result in cysteine. So what you can see is some amino acids have more than one codon, but no single codon codes for more than one amino acid. We call that we say the genetic code is redundant in that way. So amino acid CYS, the codons UGU or UGC would be the answer there. I'm not going to write it because it's too hard with this little pen thing. If we're looking at the amino acid ARG, which is arginine, we just need to find it. We can see it's there. You'll also notice though, it's down here. So arginine actually has 1, 2, 3, 4, 5, 6 different codons that code for the amino acid arginine. So for your answer, you would need to write all 6 because all 6 would code for it. So CGU, CGC, CGA, CGG, AGA, and AGG. Let's flip the questions around the other way. So question 2, identify the amino acids for the following codons. So for the codon CAC, we need to find that. So look for the C column, then the A, then the C. So CAC is there. And for CAC, we see that it relates to the amino acid his, which is histidine. So we would write down his. Find the amino acid for the codon AUG. So if we look for AUG, we can see it's down here. And it codes for the amino acid MET, which is methionine. And a fun fact for you, AUG is what we call the start codon. It's the first codon of every single strand of mRNA, which means the amino acid MET, methionine, is the first amino acid in every single protein that we have. Question 3, identify the stop codons. So if we look, we will see on a few occasions, instead of listing an amino acid, because there's certainly no amino acid called stop, we've got 3 codons that don't relate to an amino acid at all. They relate to what we call stop, which means when we are translating a piece of mRNA to make a protein, the ribosome needs to know when to stop, because a protein should be a particular length with a particular number of amino acids. This is how it knows when to stop. As soon as the ribosome reaches a stop codon, it doesn't attach an amino acid at all. It attaches a protein called a release factor, and it causes the whole process to stop. There you go, genetic code. If you're lucky, we might play genetic code bingo in class next lesson. Let's finish off this recording I'll just talk briefly through those next 2 slides. So this is just giving us a nice lead into transcription and translation. So as I said before, DNA is in the nucleus. Transcription is the process that takes the code from a gene on the DNA to make a specific corresponding strand of messenger RNA. The messenger RNA leaves the nucleus, goes to a ribosome. At the ribosome, it's translated into an amino acid sequence that forms a protein. This slide is sort of giving you a little bit more detail on that process. So a basic overview of transcription and translation. So step 1, mRNA is transcribed from the DNA. Step 2, the mRNA then leaves the nucleus through a nuclear pore. mRNA travels to a ribosome in the cytoplasm. The mRNA passes through the ribosome, and the ribosome reads the mRNA 1 codon at a time. For each codon that's being read by the ribosome, its corresponding amino acid is brought down and attached to the ribosome, and the tRNA is what's bringing that down. After that amino acid is attached to the ribosome, essentially steps 4 to 6 just continue to occur. So the ribosome reads the next codon, tRNA brings the next amino acid down. Then the ribosome reads the next codon, tRNA brings the next one down. That continues until when? Until we reach a stop codon, and then the process stops.